Okay, I think we're live. Hi everyone, and welcome to Neuro Launchpad. This is season 1, session number 3. My name is Alex Vaccaro. I am a postdoc at Harvard Medical School, and I'm also one of the co-organizers of Neuro Launchpad. I will be your host for today's session, in which you'll hear about the work of two talented postdocs. They will be very shortly introduced by Dr. Ann West, who is a professor of neurobiology at Duke. And Dr. West will get some help from Dr. Suresh Kumar, who is a postdoc from MIT, and who will be co-moderating the Q&A at the end of each talk. I want to say thank you so much to both of you for being here with us today and for taking of your time. We really appreciate it. So I see that people are coming in. I think we should get ready and get started. Please feel free to ask your questions through the chat or like the ask a question online, and we'll make sure we ask as many as possible while keeping the discussion constructive and in time. Dr. West, the stage is all yours.
All right, well, welcome everybody. It's a real pleasure to get to participate with what is a very impressive series of science. I think it's a wonderful thing, and I just would like to commend the organizers for putting this series together and really giving us a chance to hear some of the most exciting new science that's going on out there. I think taking advantage of this opportunity is great. I'm having echoes for some reason, so I'm just going to keep talking. But our first speaker, we're excited to host today Daniel Whitlet from Aubrey Samuel's lab at Harvard. He is going to be telling us today about some of the work that he has done doing connectomics in the worm C. elegans, and says he's got some access to very exciting new microscopes where he is. And so we look forward to hearing the work. Take it away, Daniel.
Awesome. Thank you so much. I'm just going to share my screen here. Hopefully everyone can see it. I'm really excited to talk today about a project that's been going on many years across multiple different labs that I started during my PhD in Toronto. So we've been trying to understand how the brain changes throughout life. Because the brain, which is the center for sensory processing, decision-making, and motor control, it really has to be highly dynamic throughout life. And this is really especially evident during development from birth to adulthood, when the brain faces multiple challenges. During this time, the brain has to support new functions, such as walking and critical thinking. It has to maintain already established functions despite a changing body. And to adapt and learn, the brain must continuously be optimized to a changing environment. It's really poorly understood how the brain copes with these challenges because of its vast complexity. So with billions of neurons interconnected by trillions of synapses, the human brain is simply too large and complex for studying the entire thing at once. Even smaller organisms such as the mouse and the fruit fly still have millions to thousands of neurons. So to study the anatomy and function of all neurons throughout an entire brain, we really have to turn to a much smaller animal, such as the nematode C. elegans, whose nervous system consists of only about 300 neurons. And so even though the C. elegans nervous system is really small, it's organized similar to the system of larger animals, meaning that the worm has a brain where most sensory information converge and decision-making happens. It has a main nerve cord where locomotion is controlled, and it has peripheral neurons throughout the body, most of which collect sensory information that's directed into the brain. So because of its small size, we can actually map the entire nervous system at synapse resolution, meaning that we can get a complete diagram of how all neurons are connected with each other. And we call this neural wire diagram the connectome. So another advantage of the worm is its stereotypic cell lineage, meaning that the birth time and location of each cell is almost identical across different animals. So we can directly compare every neuron and its connections between individuals to find out how the connectome changes from animal to animal. And then finally, sea elephants populations are isogenic because they make clones of themselves. So you can pretty much eliminate genetic differences as being the cause of any connectivity differences we observe between animals. So altogether, this really makes for an ideal system to extract fundamental principles about how and why brain wiring changes across life. We would simply have to reconstruct the connectome across all these different life stages, and then look at the changes and analyze the changes. So to do this, we developed a high-throughput serial section electron microscopy pipeline, which allows us to reconstruct the entire brain of a nematode in a few months. And I'm not sure how well this video here is coming through on Crowdcast, but what you're supposed to see is an electron microscopy volume that consists of about 5,000 images covering an entire sea elephant's brain. And here we're just seeing the images at low resolution, but each frame actually consists of more than 1,000 megapixels, meaning that we have enough resolution to clearly see individual neurons and synapses. So getting a volume like this involves staining the animal with heavy metals to make cells visible by electron microscopy, then cutting the animal into thousands of ultra-thin serial sections that are imaged one by one, and finally tracing all the neurons and annotating their synapses throughout each of these sections. So here, every neural process and muscle fiber throughout the brain has been segmented. And we focused on the brain because it contains the majority of neuron classes, and it's where most decision-making happens. So as we're going through the cells here, you'll see that they're colored by their type, which we know from their morphology and from previous studies. And we have both neurons, and we actually also have a few muscles which are directly connected in the brain. And then finally, we have all the synapses between all the different cells. So we did this for 8 isogenic animals across development, which we can see on this timeline here. If I can get my mouse, this timeline. And so with these data sets, we can examine how the synaptic connectivity and morphology of each neuron changes across maturation at the level of the entire brain. So in looking at this data, one thing that was immediately obvious to us was the brain is getting bigger without changing in overall shape. So you can quantify this by looking at individual cells, such as here's the cell called AIZ, which has a left and a right pair. And we can simply measure the length of each of these neurites, as well as the physical contact area between them. So the total length of all neurites increase about 5-fold throughout the entire brain. And this growth is proportional, such that the physical contacts between cell pairs are mostly persistent throughout life. So here, the y-axis is simply the percentage of physical contacts between cells that you see across all data sets. So for example, more than 90% of the physical contacts at birth, they're present throughout life. So the overall shape and relative size of neurons in the brain is maintained after birth. But in parallel to this neural growth, there's an extensive formation of synapses from birth to adulthood, which we can see if we plot the connectomes as network graphs, where each little circle here represents a cell, and each line represents the synapses between cells. So from birth to adulthood, the total number of synapses increases about 6-fold. But this synapse number is the synapse number increase. It's simply not because of enlargement of its existing structures. I'll actually show you that there's a profound amount of rewiring going on here at the level of individual cells to the level of the entire brain. So if we look closer at one of these connectomes, just make it bigger here. It's a bit cut off here. I'm sorry about that. Hopefully it won't have too much problems. But here we can better see individual cells, and we can see the connections between cells. And so here I define a connection as being all the synapses between two cells. So some connections consist of more synapses and are therefore stronger than other connections. So the addition of synapses during maturation creates many new connections, meaning that synapses are added between cells that were not connected before. And the addition of synapses also strengthens existing connections, meaning that the number of synapses per connection increases. So we asked whether this large increase in new connections and the strengthening of existing connections happened uniformly throughout the brain. And I'll show you that we found in at least 3 different contexts that connectivity changes throughout the brain were not uniform. So first, if we look at the creation of new synapses, or creation of new connections, one long-standing hypothesis in developmental neuroscience is that synapse formation is driven by the physical contact between 2 cells. So according to this model, neurons sharing a large contact area, such as the blue neuron here and the pink neuron, they're more likely to build new connections, whereas other neurons that have less contact area are less likely. So to determine if this was actually the case, we measured the contact area between every single pair of cells in the brain at birth that did not already have a connection, and then counted the probability of a connection appearing later in life. And we saw that a large contact area was indeed predictive of a new connection forming, so the physical contacts formed at birth appear to create a stable scaffold within which network formation unfolds. And a second form of non-uniform synapse addition was evident between neurons with different properties. So from birth, it was already evident that some neurons were more highly connected than other neurons, meaning they had more connections than other neurons, which is, I'm trying to indicate here with these 3 slightly bigger neurons. And so we considered the possibility that some neurons were simply more likely to build new synapses, in which case we would see neurons with already many connections at birth disproportionately make new synapses compared to other neurons. So this idea is also known as the rich get richer. And consistent with this idea, we found that the number of connections that a neuron has at birth is correlated with the number of new input connections they receive later in life. So here, every data point is 1 neuron. But surprisingly, neurons with more connections at birth were not more likely to add new output connections. So new connections only converge onto these highly connected neurons. And similarly, neurons with more connections at birth are more likely to disproportionately strengthen their existing input connections, but not strengthen their output connections. Thus, maturation focuses the flow of information onto these most highly connected neurons at birth. Or said another way, highly connected neurons become better integrators of information, but not broader communicators of that information. And a third form of non-uniform synapse addition is evident when comparing the relative strengthening of neurons' inputs and outputs. So we correlated the number of synapses for inputs to the same cell, outputs from the same cell, and connections between different cells. So I'm going to show the result in this plot here, where the y-axis, it sounds a bit scary, the coefficient of variation in synapse number, it simply means how different the synapse number is between 2 connections. So over time, unsurprisingly, connections to and from different neurons, they diverge in synapse number. They're all growing, but they're growing at different rates. The same is the case for inputs to a cell. But interestingly, the relative strength of outputs from a cell are maintained, meaning that the outputs grow at similar rates. The contrast between inputs and outputs is most clear if we just take the difference between the 2, which is shown down here in this plot. So the relative strengths of outputs but not inputs are maintained across maturation. Or said another way, each cell regulates the strengthening of its own synaptic outputs, but does not dictate the relative strengthening of its inputs. So in summary so far, the maturation of the connectome is not simply uniform, but it follows certain principles that are evident at the level of individual neurons. And here I'm showing the 3 I just talked about. But all these principles here, they're derived from overall statistical analysis of anonymous neurons. And one of the advantages of using C. elegans is that we actually have the identities of each of these cells. So you can examine exactly which specific connections change. So to do this, we classified connections by their stability across individuals and development. So you're seeing a toy circuit at 3 different ages. And some connections, the one shown here, they're stable, meaning that we see them in every single animal. So some do grow a bit stronger, but that's expected because we're getting a large addition of new synapses. But other connections, they're developmentally dynamic, meaning that they show a statistically significant increase or decrease in synapse number, different from the rest in a very stereotyped manner across maturations. So these connections sometimes even form new connections, which is the case between these two neurons here, or they eliminate existing connections, which is the case for the connection here that disappears. And then there's other connections that are variable, meaning that they exhibit no consistent trend in their changing synapse number, and they're not present in every animal. These variable connections are typically only present in 1 or 2 animals, and often not animals of the same ages. So if we count the number of connections for these different 3 types, we see that variable connections are surprisingly common. In the adult, they make up almost half of all connections, and about the same number of the stable connections. Although these variable connections, they're typically weak, consisting of fewer synapses than other connections. So if we look at the total number of synapses, it's a bit hard to see, synapses that make up these variable connections, the proportion is smaller. But variable connections still make up about 15% of all synapses in the brain, so they cannot simply be ignored. And this degree of variability really contrasts with the widely held view that the C. elegans connectome is hardwired. So we considered the variability is more prominent during development than in the mature connectome, in which case we would have overestimated the prevalence of variable connections. But if we just look at our 2 adult connectomes and compare the variability between those 2, we get similar numbers. So one thing that's indicating that these variable connections have some importance is their prevalence. Another is that they're not simply randomly distributed throughout the brain. And we can see this if we look at the connectome and here classify cells into different types. Types here being sensory neurons, modulatory neurons, interneurons, and motor neurons. And then we can simply compare the number of variable connections between each of these cell types versus the non-variable connections. And if we do this, we see that variable connections are most common among modulatory neurons and least common among motor neurons. The high stereotypy of synapses from motor neurons may reflect the requirement of high fidelity in circuits for motor execution. On the other hand, modulatory neurons, which can secrete monoamines and neuropeptides by volume release, may have the weakest requirement for the precise positioning of their synapses because they can exert long-range effects. But this non-uniform distribution of variable synapses suggests that variability is in some way regulated and may be functionally important. And this makes sense because even in highly isogenic populations, behavior is variable. This is clear when giving the C. elegans the choice between 2 different odors, for example. So we can see that here where I'm going to play a video in a second where we place a bunch of C. elegans on one side, and then we put an attractive odor indicated that there's food on the other side. And if this plays, you'll see that over time, the majority of worms in the population moves towards this odor in search for food. But there's always a certain proportion of the population that consistently makes another choice and moves the other way. This behavioral variability is advantageous to the population because if suddenly the attractive odor becomes dangerous to the animal, the entire population would die out if they all behave the same way. So behavioral variability is not random, and it's highly regulated. And since wiring variability, since wiring variability is not random either, it could be the source of this behavioral variability. So with this in mind, it also makes sense that motor neurons have the least amount of variability because the worms want there to be variability in the decisions they make, such as should I or should I not crawl towards this odor, but once they've made a decision, it should be executed faithfully by the motor neurons. So variable connections are likely important. And similar how we examined these variable connections, we ask if developmentally dynamic connections, which are the ones that change across maturation, are evenly distributed throughout the connectome. So we again group cells by their type, this time visualized a bit different as a network graph, where each arrow indicates all the non-variable connections going from the cells of one type to another. And we found that developmentally dynamic connections, they're distributed throughout all layers of the network, but it is most changes are seen at the periphery, from sensory to modulatory to motor neurons. And in contrast, the communication within the interneuron layer here is remarkably stable. This stability of the core parts of the nervous system across maturation implies that the central processing unit is robust enough to be used in different contexts. Maturation changes the flow of sensory information into the central processor and changes its readout without changing the central processor itself. So we've observed patterns of synaptic change, which are evident at the level of individual neurons, as well as patterns evident at the level of neuron types. And finally, we wanted to ask if the collective set of synaptic changes leads to changes in information processing at the level of the entire brain. So I don't have much time left, but I'm quickly going to go over two different ways we looked at that. First, we examined the directionality of information flow. So we classified connections from the sensory towards the motor layer as feed-forward, connections from motor layer to sensory layer as feedback, and connections between neurons of the same type as recurrent. And a network with more feed-forward connections is thought to be more reflective, reflexive, with a certain input consistently resulting in the same output. So interestingly, we found that across maturation, stable feed-forward connections were preferentially strengthened more than feedback connections. And additionally, new connections added during maturations, they were more likely to be feed-forward compared to stable connections. So together, this results in the connectome becoming increasingly more feed-forward biased. So one global pattern of brain maturation increases the signal flow from sensation to action, making the brain more reflexive and less reflective with age. And finally, another global pattern of brain maturation was evident when we examined the community structure of the brain. So we used weighted stochastic block modeling to group neurons of similar connectivity into distinct modules. And this says cells per module. We found that the connectome becomes more modular, increasing from 2 or 3 modules at birth to 6 modules in the adult. So synapse formation progressively increases the community structure of the brain as subnetworks for sensory and motor processing gradually emerge with maturation. And interestingly, these subnetworks, they're actually spatially organized in the brain as well, reminiscent of brain lobes of large animals. So to sum up, we've uncovered principles of synaptic remodeling on multiple levels of the connectome. At one level that applies to individual neurons in the brain, we've observed patterns of synaptic remodeling that alter the number and strength of individual connections. And at a second level, we've observed synaptic remodeling that differ between cell types. And at a third level, we've observed network changes that altered the directionality of information flow and the segregation of information processing throughout the brain. And before I end, I need to stress that a lot of people were involved in this project. I want to especially thank my old lab, Mayzen, and then the Samuel and Lishman labs, but there's also many others.
Fantastic. That was a beautiful talk, Daniel, and the video played great. It was very cool to see that develop. I had a question for you, and please enter your questions down here in the question box. Suresh will be watching for them, but I wondered if I could kick it off and just say, I was waiting for this. I'm always the one who says, the worms, they're so stereotypical. Everything's always in the same place. But I was sort of curious about that as you're doing your connectomics. You mentioned variability, but are you actually doing connectomes of multiple organisms? What was variable? What did it mean for a synapse to be variable in your story? Was that between 2 organisms, or was that between one cell type that you found different synapses?
So we did 8 animals, and if we wanted to perfectly measure variability, we would have to do many animals at the exact same age, so we could eliminate developmental differences, and then basically see which are variable. But obviously, we don't have that data, so all we can do is say across development in these different animals. But
then what differentiates variable from developmentally differential then? Because you had those as 2 separate categories.
Right. So developmentally dynamic, they show a clear pattern. So across development, they show an increase in synapse number that's statistically significant, or a decrease in synapse number. Whereas variable connections, they just often appear in one animal or two animals, and they're sort of random. There's no clear pattern to them. And even if we look at the animals we have that are the same ages, they show the same amount of variability.
Are these males or hermaphrodites, and does that make any difference? I was thinking if you could have sex as a difference, that would be a good way to go looking for differences in development.
These are all hermaphrodites, because there does seem to be a lot of differences between sexes as well. So we wanted to completely eliminate that. Yeah, we wanted to make the animals as similar as possible, so they're isogenic, they're raised in the same environment, and yeah, trying to really eliminate all variability from the environment, although we can't obviously eliminate everything.
Are there questions out there that are coming in, or from you guys?
I have a question. That was an excellent talk. I have a question about gap junction synapses. So for some reason, there is a resound. I will continue talking. So do you see any change in gap junctions or electrical synapses throughout the development?
It's a good question. And so what's tricky about gap junctions is that they're really hard to objectively identify by ultrastructure alone. And so often you can say if there is a gap junction, like a very clear gap junction, you can see, oh, there's definitely a gap junction here. But it's harder to say, oh, there's definitely not a gap junction here. Like you'll end up having a lot of false negatives. And that makes it really difficult to analyze with certainty whether there's a change across development, because you'll basically just have a bunch of holes. The only way you can be 100% sure if two neurons weren't making contact whatsoever at one point, and then you see a gap junction later. So for all of our analysis, even though we did annotate the electrical synapses when we were looking through the data, we didn't include it in the analysis just as a measure of caution. But there does seem to be some, I mean, knowing just if there's definitely a few of them that does change, but I can't say if there's any like big patterns or if they follow these same principles we've seen for the chemical synapses. And
do you see any gap junction subunit change, like connexins or pannexin subunits being like change from stage to stage?
So we haven't looked at that because we're just looking at the ultrastructure. As far as I know, we can't tell which from ultrastructure alone what subunits there are. I mean, it might be that's an interesting question. I think someone should look into that. But I think others have shown that there does seem to be, if you changes in expression of different gap junction proteins, especially I think from Oliver Hubert's lab, did like a big screen of all these different things and saw that as well when the worm enters like a stressed period of life, when it goes through like if it's starved, that also seems to change the expression of these gap junction proteins. So there must be a lot of dynamic expression and function of gap junctions. Yeah, it's just we have to optimize how we can really classify them and identify them before we can really answer those questions. Okay. And it's something my old lab makes, and they're working on that. Yeah, it's just not finished.
We get two other questions in the chat here. One about the variable synapses asking if they go in a particular direction, do you see clear evidence of synaptic pruning in specific circuits, so the numbers going down?
Yeah, so it depends what you mean, how you define pruning. So you can either say pruning is when you're definitely seeing synapses disappearing, or it's just if you look at the relative number of synapses, then it's decreasing, because it's definitely the case that there's sort of a massive addition of synapse number, and there's some connections that don't seem to get new synapses. So relative to the other connections, they're definitely being pruned. Which happens a lot. The other case of where you see synapses actually disappearing, it's rare, just because you're getting so many new synapses added, but that's definitely the case as well. It's rare.
And then Chin has our last question here. She says, do you have any behavioral data from these animals prior to the imaging?
I would love to have that, but no. The EM processing is very low throughput. So right now we have to do like hundreds of animals before you actually get one that's a really good sample that you can then reconstruct. And but yeah, that would be awesome if you could really do behavioral data or even whole brain imaging on an animal, then afterwards reconstruct its connectome. So you can then you could directly ask that question, right? Is it because of the wiring variability that you see behavioral variability? Yeah.
Well, fantastic. Thanks, Daniel. Big round of applause from everybody. All right, great. All right, so on to our second speaker here, Xin Jin, also from Harvard, also with a history of working in worms, however, has seen the light and has gone to, no, just kidding, has gone to a much larger sort of organism, otherwise known as a mouse. She is a junior fellow at Harvard, which if for those of you who might be on the this call who haven't experienced that position before, if you want to know more about it, Xin would be a good person for you to talk to. It's a very unusual sort of free postdoc where you're kind of in charge of your own game. And she has taken advantage of the resources in her environment to do a project that's joint between Paula Arlada's lab, looking at cell types in the nervous system, and Fang Zhang's lab at MIT working with CRISPR to perturb genes. And she's going to tell us a little about that today.
All right, thank
Take it
you
away.
very much. Thank you so much, Anne, for the introduction. I also want to thank the organizer for putting together this fabulous seminar series and for the opportunity for us really to share our work here during this very special time. As an ex-worm person, I really enjoy my panel speaker, fellow colleague, Dan's talk. It's an honor to be here with you. So as Anne introduced, I'm going to talk about this project that is done in Feng and Paula's lab with close collaboration with Aviv Regev. And we're interested in developing a systems approach to study gene function inside an intact tissue. And this idea is really driven when we think about neurological diseases and disorders. In the past almost 2 decades, we have already learned a lot about how many of the diseases and disorders are strongly implicated with risk genes and loci. And particularly for autism, we already know a lot of genes that's strongly implicated. For example, one of the syndromic cases of autism called Rett syndrome is caused by the mutation in MACP2, which is a transcriptional repressor activator that is required in both development and adult stage. And another set of genes, for example, SYNGAP, is a gap protein that operates at the junction of excitatory neurons and excitatory synapses. They are actually part of a very large glutamate receptor complexes and regulate receptor trafficking and neurotransmission. I will give you one last example, which is Fragile X, implicated by the mutation in gene called FMR1. And this time, FMR1 is regulating protein translation, among other things, and other biological processes. So we could go on and on about these single genes that are strongly implicated in autism. And these are actually de novo loss of function variants, which means that they have very large effect sizes and often highly penetrant. And these are actually the starting point as an entry point for us to understand such a complex disorder such as autism. And these single gene studies in the past have really been important because they set a foundation for the entire field. They help building the important models from vivo to vitro. They allow us to actually do experiments in the electrophysiology, molecular biology, and behavior aspects to try to sort of approach this complex disease. But we should also realize that through more and more human genetic and sequencing are done, such risk gene list is also growing. And in the recent cohort study that was just published earlier this year, it is shown that there are at least hundreds of such de novo variants present across genome. And as you can see on the y-axis, you can interpret it as a statistical significance for each of the risk genes. And on the x-axis are basically these genes spanning across all the human chromosomes. And some of these genes might be familiar to you, and some of them don't actually have mouse model or cell line made yet. The nature of these genes and protein are very different. They might operate at many different biological processes. So just based on the nature of the genes and the protein that it encodes, we really don't know in a priori what could be the common mechanisms behind. So the question is, are these hundreds of risk genes that I just presented to you actually affect hundreds of different things? Perhaps they are probably in many different cell types, for example, affecting different biological pathways, and therefore they would deserve hundreds of very independent studies and mechanisms when we are approaching them. Or alternatively, is it possible that these genes actually could be converged into subtypes? By converging, I mean, is it possible that some of these autism risk genes will affect the same cell type at the same developmental timing, and even maybe some of them affecting the same molecular pathways? And this is really important because it's telling us that some of these risk genes, if this is true, could be grouped for mechanistic studies and therapeutic considerations in the future. And to sort of answer this question, it really demands a systems genetic approach to study the loss of function of these risk genes. And it's important to do this in development, in the context of a developing utero brain. So we want to establish this kind of genetic screen because making knockout and studying individual knockout is very time consuming and costly. We need the scalability for the number of genes that we're studying here. So in our design, we want to focus on development. We want to inject the libraries of genetic modulation, in this case is CRISPR loss of function, very early in neurodevelopment, and analyze the postnatal brains. And we want to focus on identifying what are the cell type that's most vulnerable, what are the cell state changes that's related to the loss of function. We make the assumption that we want to study the sort of the mechanism and as well as the molecular underpinning by perturbing these genes in development when very dynamic cell fate determination and cell maturation is happening. And to build this system, we really stood on the shoulder of the giants who developed in vitro perturb-seq. We use very similar molecular approaches. And the whole idea of this method is that instead of analyzing individual animals and treat them as individual sample, we want to actually individually analyze clones or individual cells. That would really drastically decrease the number of animals and number of experiments that's involved. So we inject a pool of individually packaged lentivirus, and each lentivirus vector will perturb one and only one autism risk gene, or a negative control. We inject this pool of lentivirus very early at embryonic day 12, and this is the time that the brains are not fully formed yet. And you can see that this lentiviral infection will sparsely label, in this case, the blue epithelial cells at E12.5. And all the cellular progenies that are coming out from this labeled cell will also carry the fluorophore expression, as well as the CRISPR perturbation for the given gene. At postnatal day 7, we can use FACS to enrich for these blue perturbed cells and use single-cell transcriptomic to analyze their transcriptome. And the single-cell resolutions is really required because this allows us to analyze the perturbation of many genes at the same time in this mosaic brain, and only computationally deconvolute for each of the cells what are the perturbations that's encoded by reading out the barcode from the lentivirus. And ultimately, our questions, what are the cell types and molecular pathways that's most affected, as well as convergent across this cohort of autism risk genes. I want to emphasize that this method is highly efficient because it allows you to look at many different perturbations in one experimental batch in one brain at the same time. And precisely because of that, this method is less noisy or less likely to be biased by other confounds such as sequencing depth, developmental timing, and therefore is actually more robust, allow us to look at more subtle changes in the transcriptome. But there are also some features that I want to mention in this particular design. So, for example, we're injecting guide RNA in vivo, and therefore this is only a result after analyzing a mixture of homozygous and heterozygous loss of function. We did test our guide RNA to make sure the gene knockout is efficient. And also, this is not really resembling a patient state, because the mouse brain in this case is actually a huge mosaic, and only less than 0.1% of the cells are actually perturbed. And they're actually sitting in a sea of wild-type, normally developing brain cells. And this method allows us, because of this, to focus on the cell autonomous and cell intrinsic effects. You can think about this as more like a conditional knockout that people use in the traditional mouse genetics to study gene function. And ultimately, of course, all the functions and readout that we have is acquired from a developing mouse brain, which might be different from the human genome context. And also, we decided to introduce the perturbation at E12 and harvest at P7, and that also limit the molecular readouts that are happening during this time of the development. But in the future, we could introduce perturbation and harvest at different timing to acquire more developmental granular insights of how gene function. So from this sets of studies, we collect in total 46,000 cells out of 163 embryonic injections. And we can first cluster all these single cells into generally cardinal cell types. And unsurprisingly, we can identify 5 major cardinal cell types from the new cortex. As predicted, this is what the early injection and labeling allow us to target. And these are the cells including neuronal cells as well as non-neuronal cells. And we can also check that in a mouse coronal section brain, this early injection of lentivirus allow us to target not only the cells in the cortex, but also in many other brain regions, such as hippocampus and thalamus. And in the future, although we only focus on cortex in this study, we could use in vivo perturb-seq and then similar viral delivery method to study the neural and glia development in other brain regions and at different developmental timing. So I mentioned that we can identify 5 major cell types in the cortex at this stage, but we can also take advantage of the single cell data nature and subtype these perturbed cells. For example, we can take the excitatory projection neuron out and further classify them into subtypes. And in this case, we can classify them into several subclusters that correspond to their laminar positions in the cortex. This is important because this is really showing that the resolution of our readout in the perturb-seq is much better than the traditional bulk approach. For example, if perturbation affects only 1 out of the 5 cardinal cell types, or if the perturbation affects one of the subtype of the projection neuron that's showing you here instead of other subtypes, the perturb-seq single cell readout will be captured. That may have been missed if you were doing bulk sequencing or bulk analysis traditionally. So using the machine learning-based topic modeling and the gene network analysis, we can extract gene programs from all these single cell data. When I mentioned these gene program or gene modules, they're actually networked of the genes that covary in the data set, which we are using to score the phenotype upon the perturbations. So some of these gene modules describe the specific cell types, such as the PN1 gene module that I'm showing you here on top. As you can see that a lot of these expressions are highly correlated to this corner on the T-SNE, which correspond specifically to layer 5. So we believe this module represents certain features specific to a subtype. But this is not all the modules that we are getting. For example, in PN2 module, and this module represents neurite development, which is actually concerned biological processes that's present in all the different subtypes. And as you can see on the T-SNE feature plot on the right, the expression of these PN2 modules is actually seen in all the subclusters. So we do have both of these 2 kinds of subsite-specific or feature-specific gene programs analysis. So why do we have to use this gene program as a phenotypic vocabulary? This is because we know that many biological changes operate as networks, not just single genes. And we assume that if the autism risk gene perturbation indeed change some kind of network property, physiological property, there must be network changes in the transcriptome, such as a list of molecules go up or down together. And also because gene program is superior because it offers a more robust readout compared to the single gene traditional differential gene expression analysis. So this is what's sort of described before comparing the traditional gene level analysis with the gene program analysis. In the top right corner, it is a data set taking 1,000 blood monocytes and perturb them with LPS injection. And we expect to see the transcriptomic changes associated with inflammation. But now we can use this data set and exercise downsampling. We can downsample on the x-axis, which is reducing the number of transcript included in analysis, or we can reduce on the y-axis, which is reducing the number of cells included in analysis. And as you can see, when we go down on the y-axis, we expect, as expected, we can see the correlation compared to the top right corner gold standard has dropped pretty quickly. However, if we do the similar analysis, but now using gene program as a readout, you can see that now we can actually fairly comfortably subsample to about 20% of the cells included in the whole analysis and still obtain a fairly high phenotype correlation. And this is, again, showing that gene program can allow us to use much fewer cells, such as about 30 to 50 cells per perturbation, and obtain very robust perturbation. And personally, I like gene program because instead of 1 or 2 genes that we want to read every single paper about and to guess what our biological implications are, gene program is more interpretable and gave us sort of more clues about what potentially is going on at a biological level. So now I just want to show you sort of the general landscape view of our results here. So each row is showing you one of the gene programs that belong to a specific cell classes. For example, PN1 is the gene module number 1 extracted from the projection neurons. And each column is autism risk gene perturbations, and we total analyze 35 autism risk gene perturbations. So even in this 35 gene perturbation, we already observe some level of recurrent phenotype. And some of these effects are quite cell type specific. For example, perturbation of the gene called ADNP is actually very specifically affect projection neuron 1 module in the excitatory neurons. But some of them will hit multiple module in multiple cell types. So this is really the power of single cell analysis, allow us to capture the phenotype that could have been masked in the bulk analysis. And interestingly for me, I was surprised to see that both neurons and glia can be directly affected in this panel of perturbations. Many of these risk genes actually express very broadly early in development. So it is really interesting to think about in early time points, what are the direct impact of some of these risk genes, and which will guide us to think about how to build better vitro models to study them. So this is really a very rich data set. Just like all the genetic screens, you might wonder what these hits I just showed you means and what have we really learned from these results. Now I'll go through some of the key phenotype that I found interesting, but before diving into a detailed set of validation experiments at the end of my talk. So what have we really learned? We found out that there are generally 4 cell types that are affected. That's projection neuron, interneuron, astrocytes, as well as oligodendrocytes upon the perturbation of autism risk genes. And I don't have time to show you the data, but we actually co-analyze our panel of data with the human post-mortem autism brain data sets. And these are also the cell type that seem to be affected in the human post-mortem analysis. And particularly, we found out that layer 4 projection neurons seems vulnerable in development upon the perturbations. And this is also the time that the thalamus, which represents the inputs, being wired into cortex. So we suspect that this might be linked with the abnormalities in sensory processes that many have studies in the mouse model, at least. And secondly, we also noticed that ANK2 perturbation actually is associated with upregulation of a module in the interneuron and specific subtype of interneuron expressing NDNF. And in this phenotype, we observed the upregulation of many synaptic proteins, including GABA receptor as well as ion channels. And we suspect it's related to anchoring's role in connectivity. Especially ANK2 is known to have a role in excitatory pituitary neurons and regulating their connectivity and axon guidance. And this is a new role in interneuron in probably an early time in development at P7 in a very similar pathway. We also observed that the protoplasmic subtypes of astrocyte, there are 2 gene signatures seems to be altered upon the perturbations. In the rest of the talk, I would just tell you one example that we did a sort of validation using a germline mutant animal. And this is a phenotype regarding a glial cell type called oligodendrocyte. So oligodendrocytes is a glial cell type that actually take a really long time to mature. At this stage, postnatal day 7, the majority of the oligodendrocyte in our data sets are actually progenitor cells, labeled here in purple and red. And this ODC1 module that I showed you seems to be highly expressed in sort of like the north part of the plot, which resemble the progenitor's signature. We found out that perturbation of 2 autism risk genes, that are CHD8 and GATA-D2B, lead to decreased expression of this ODC1 module. And CHDA is a very interesting gene. It's a chromatin modifier and can recruit histone protein to negatively regulate Wnt signaling. So at the same time, when I saw this result, there was a paper showing that CHDA can be a chromatin modifier to interact with OPC maturation genes through the ChIP-seq data, which really gave us confidence. And we went on, used a mouse model that is heterozygous mutant of CHDA to validate some of these results. So first, to directly validate our result from the perturb-seq, we took the same genes and the similar markers from the oligodendrocyte precursor subtypes and used in situ hybridization to quantify the gene expression in situ in the layers of the cortex at postnatal day 7. So we observed that 2 of the markers, PDGFR and CSPG4, both of them showing about 20 to 30% reduction of the gene expression level in the mutant. And this is the subtle changes that correlates well with what we observed in perturb-seq. We also found out that oligodendrocyte in the upper layer or deep layer doesn't have a huge difference in this change of expression. And we suspect that this downregulation of the immatureness might sort of resemble some kind of changes in the maturation timing. So we did a converse experiment, which is we took the animals from postnatal day 11, when it's a little bit later in development, and we used immunohistochemistry to examine the expression, the oligodendrocyte matureness. Because after analyzing RNA throughout this project, I sometimes wonder whether at the protein level there's any differences. So we did a immunohistochemistry at multiple time points. Here I'm showing you at postnatal day 11, where we observed upregulation of myelin basic protein MBP in the cortex, suggesting that there's advanced differentiation of oligodendrocyte, at least at this time points. So because oligodendrocyte is really a relatively rare cell type at such an early time point of development, without the single cell data, we really wouldn't have known to focus on oligodendrocyte in development. So now we know the maturation and differentiation is affected in OPC, but there are really just more to learn, such as how these differences could possibly lead to the physiological behavior differences, both at the level of the differentiation as well as the final myelination. These are processes that's very vulnerable and tunable from activity and experiences. And this is also a way for me to think about how non-cell autonomous fact can come into play. The directed fact may be in oligodendrocyte, but oligodendrocyte or the risk gene itself doesn't manifest nervous system and behavior abnormalities. They really need to sort of like impact the neural connectivity or the circuit refinement to change the function of the brain, which is a future direction that we really want to focus on. So just to summarize, we described to you an experimental approach that is the generalizable approach to study gene function at a scalable way. From there, the single cell really unveiled the biology that could have been masked by bulk sequencing, and there are so much rich insight that should be followed up in the future. And this also allows us to discover new biology that these risk genes are, on the one hand, represents what the disease might be happening in the brain, but at the same time, I think thinking about these risk genes are actually the key entry point to study fundamental biology, because gene itself, autism risk gene itself, do not really give rise to thoughts or behavior. It's really the nervous system that does. So Perturb-seq allow us to take these genes to understand what are the cell type and molecular pathway that they affected, and to understand how these molecular components actually operate on the nervous system to exert function or dysfunction. So I will spend maybe one slide talking about ongoing effort to continue developing function genomics, because we know that clearly there are a few points I didn't mention in the first Perturb-seq project, which is first, mouse and human neurodevelopments are very different. Human has more neurons and much expanded upper layer cortex that's recently expanded in evolution. Is there any human-specific feature that we are now really capturing in the mouse experiments? And second, by dissociating these cells in the brain, we really lose the spatial readout of where these cells are and who their neighbors are. So in the future, with Paula's lab, I set up Perturb-seq in human cerebral organoid to study the human neuron differentiation in these 3D cultures. And we collaborate with Xiao Wang, perform star map that allow us to use spatial transcriptomics to read out the transcriptome as well as where these cells are relative to each other. Now we can do Perturb-seq to ask how are these altered gene program location or even their cellular environment can alter the manifestation of the perturbations. Okay, so the last but not the least, I want to thank Paula and Feng, my mentors, without whom I simply won't be here. Aviv has been a key collaborator throughout this project, as well as Joshua Levine. I work very closely with a computational biologist, Sean, really taught me a lot about analysis, and we did many of these fun projects together. Michelle and Amy helping me throughout this entire project. And I should also say that this project wouldn't have been started without Elise Robinson and Mark Daly, who are genius human geneticists who taught us about these disease risk genes, as well as my funding sources, and I'm happy to take questions.
All right, that was great. Hey, Tin, I have the same question actually as AJ, what he put here into the chat, which was when you showed that big plot and you had all your genes across the bottom and your different cell types across the side, you mentioned, I mean, you guys can't really control if these are heterozygous or homozygous mutations, and that's a big deal. In humans, these mutations are almost only heterozygous and not homozygous mutations, not even necessarily all loss of function. So, so what AJ was asking is he says, I know this is difficult to resolve, but you expect transcriptomic effects of mutations. Do you expect them to be the same with hets and homozygotes? And I would ask you, do you know for each individual cell, whether it's a heterozygous or homozygous mutation, and do you see the same perturbations with both?
Yeah, that's a great question. That's also the question that we keep asking ourselves throughout the entire project. So a few technical notes. Controlling heterozygosity using CRISPR screen is possible. It would just be very challenging. So one sort of creative way to do it is you can take advantage of the naturally occurring SNPs. And you can have these hybrid sort of animals that, and then you design guide RNA specific to one allele, not the other. I actually try out some of it, but a lot of these highly SNP, sort of highly variable mouse strains are very difficult to work with, though the breeding was not very easy. But in the future, it's doable. It's just a technical constraint. So that will be the most of the beautiful experiments. In terms of can we see it from the data, not from the current data set. We try to do many things, try to see if we can, take the same perturbation in the same cell type that's reasonably homogeneous and try to cluster it into. It doesn't seem to be the case. And regarding your question specifically about how the homozygous and heterozygous will have different function, absolutely. Sometimes I even thought, is it possible that homozygous might have a much stronger phenotype that we might have already lost it during development, or we might have already lost it when we're doing FACS. And that's the catch-22 of genetics, right? Like the most interesting phenotype may be the one that's already lost, and that's the one you're now seeing. So I think to a certain degree, but going back to the question, I think the in situ Perturb-seq might be able to help out a little bit, because we're not fax purifying these cells, we're not adding this purifying selection pressure, we may be able to design probes to specifically looking at these allele to check the heterozygosity in the in situ sequencing. So that's actually exactly what I'm trying to improve. Whereas this experiment I told you was done in 2018, I think 2017, 18. At that time, we were using a version of 10x, and gene capture rate was obviously not as good as what you could get today. So yeah, that's a very important question. That's why I feel like we have these results, and to follow up, to look at it from a different system, using a different experiments even, making me sort of more comfortable if we know why and how, and mechanism is important.
You know, just since I made a derogatory joke about worms earlier, I should say something nice. You know, in worms and flies, oftentimes the best mutants are actually the hypomorphs, because you don't get compensation for the loss of the gene. And so you could create a hypomorph with dead Cas9, with dead Cas9 crab. So, you know, doing CRISPR-I instead of actually doing CRISPR knockout. Did you guys ever try that?
Yeah, totally. So that's why in one of the slides, I put it like a general scheme of in the future, how do you want to do it? I think bidirectional control and also, you know, quantitative control would be really nice if we can control the dosage at the same time examining the phenotype. I am not aware of people doing in vivo CRISPR ANI as of yet. But I think the animal is probably, like, there's a some-type animal transgenic mouse that's being made. So I don't think it's not it's not sort of like established. Probably we need to be a little bit careful about, you know, quality control, because CRISPR AI might depend on, you know, the chromatin states of the target gene. It may not work efficiently, and during development things might, you know, dynamically change. So there are a few things I would probably, you know, want to quality control a little bit more. But yes, that's totally the approach I'm hoping to sort of get people excited and get people to work on.
The mice exist, so you're not surprised to hear that. Do you guys have questions? Do you guys have questions? There aren't any other in the chat yet. Suresh or Alex? It's absolutely gorgeous work.
Thank you.
Yeah, and a ton of beautiful, beautiful data. All right, we'll turn it back to our host.
Yeah, thank you so much to both of our speakers. These were amazing talks. Thank you to our moderators, Dr. West and Suresh. It was really nice to have you on stage. I hope you guys will join us next week, same time. And yeah, like, see you, I guess, around. And again, thanks to our two speakers. Good job. Bye.
Bye.